The second line of defense is more sophisticated than the first line of defense because the immune system should actually do something to deal with the pathogens that have penetrated the first line of defense. You want to imagine the second line of defense as being a combination of the FBI and the armed forces involved in combat or rescue. We're going to classify the second line of defense into three types: protective processes, protective proteins, and protective cells. So we'll start with the protective processes. There are two types of protective processes that we will look at. One of them is inflammation, and the second is fever. Both inflammation and fever are beneficial to the body unless they go out of control. The second is protective proteins. Let's look at two types of protective proteins. One of them is the interferon, which is an antiviral protein, which we will look at closely in a bit. The second is a complement protein. Complement proteins are a set of proteins floating around in the blood, waiting to be activated by the presence of a pathogen. The third group is the protective cells. We will take a look at two major protective cells at the second line of defense, and they are the natural killer cells and the phagocytes. The natural killer cell is a kind of lymphocyte, and the phagocyte is the macrophage or monocyte. A natural killer cell is a special type of lymphocyte that has the ability to carry out immunological surveillance. These cells travel through the bloodstream looking for cancer cells and virus-infected cells. Unhealthy cells such as these look quite different from the normal cells. These enemy cells carry special receptors on their cell surface that can be recognized by the natural killer cell. So let's take a look at how a natural killer cell destroys the enemy cell. The natural killer cell is shown here in pink, and the enemy cell is shown in blue. The enemy cell could either be a cancer cell or a virus-infected cell. So when the natural killer cell comes in contact with the enemy cell, it releases proteins known as perforins. Perforate means to make a hole, and perforin is a protein that makes a hole. So that's exactly what it does. These proteins reach the enemy cell and make a hole in them. The hole is then used by the NK cell to introduce enzymes. These enzymes are small and granular, and so they are called granzymes. These granzymes make the enemy cell undergo apoptosis, meaning it causes the enemy cell to commit suicide. Once the enemy cell is destroyed, it is then cleaned up by a macrophage, which carries out phagocytosis and clears the debris or remains of the destroyed enemy cell. Now let's see how a phagocyte destroys a pathogen and presents its antigens to more sophisticated cells of the immune system. So here is the pathogen in yellow and its associated antigens. You want to imagine antigens as if it were buttons on the coat of the pathogen. Here is the macrophage attempting to engulf the pathogen. We call this process endocytosis, or more specifically phagocytosis, because the cell is actually eating the pathogen. Once the pathogen is inside the cell, it is put into the lysosome. The lysosome then destroys the pathogen while keeping its antigens intact. These antigens, imagine they are buttons, They are worn by the phagocyte to say, I've just killed something, and here are the buttons to show for it. These buttons are the antigens, and these antigens are presented to T lymphocytes, which are the most sophisticated type of white blood cell in the third line of defense. So the antigen presenting cell, or APC, is nothing but a phagocyte.